Hello there, and welcome to a brief overview on the stages of scientific investigation. Now, with scientific investigation, there's always a general set of steps. I'll back up a page here. General set of steps. There's an observation, hypothesis. Sometimes prediction is put before the hypothesis, so don't panic if you see one in front of the other. The flow is still there, and keep in mind it's so adaptable depending on the type of science that you're practicing and with the type of research. Are you looking for just a specific drug design and development? Are you looking for the effects of global warming? There's a huge range in the types of research and the scale of research. It can a little bit of a change there. Also, there are predictions. So again, sometimes predictions becomes before hypothesis, but they're connected together. And then there are testing. Testing, again, can be fleshed out into everything from analysis to research to design. So this book kind of goes more the big categories here. I'm going to talk a little bit about some of the aspects of testing here at the end, because that is an area students get just need to kind of get that sorted out and explained a little bit. And then there are typically controls associated with the test that's there, and I'll go through that. We'll talk about variables and dependent, independent variable. And if you're taking the lab for this class, great information for that as well. And then finally, kind of your conclusion. Was your hypothesis accepted or rejected? And we do this on a very sort of casual level, kind of on a day-to-day basis. Maybe we don't put in all the controls and variables, and so that's kind of maybe what separates out, quote-unquote science from what you do. But certainly you probably have a hypothesis about how long things are going to take you if you drive and you're in traffic, or you're on a bus, how long is it going to take you to get there? Is the bus going to be on time? Is your ride going to be on time? What's traffic going to be like? Is it a holiday? Is it the summer? All these sort of aspects go into your prediction as to maybe your travel time. So we do this all the time, and that's just one example of many that are out there. So I did want to talk a little bit about some of the items of testing. So you have different variables. So you have what are called controlled variables. I'm going to actually type here, so I think I can fit a little bit more in here. So we're going to do it that way. So bear with me 1 second here. And controlled variables are variables that stay the same throughout an experiment. So if, for example, drug studies are fairly easy because those are small, very not small scale. They can be large scale, but small in terms of you're only testing one parameter and you have a little bit of control over what your variables are. So for example, in a drug study, might be things such as dosage, health of the patient. Sometimes we try to control for lifestyle, especially if it's something that looks at like blood pressure or stress levels or cardiac health, that sort of thing. Gender, age group, all of those things kind of can tend to stay the same or well defined. Then we have what's called independent variable. And so it's not that these are so particularly difficult for folks to understand, but sometimes you just get the terms switched around. This is the item that is tested. So what is altered between groups? We'll talk about the groups here in a second. So there, in a classic setting, there's a control group, doesn't get the independent variable, and so sometimes this can be in a drug study a placebo group, because sometimes people, report not feeling well if they don't think they're getting their medication. So sometimes a placebo is given, but then we found this amazing thing called the placebo effect. Then there's an experimental group, and typically with drug studies, you might have multiple experimental groups. Maybe there you would have different dosages and different ages. So some of those would change in your controlled variables a little bit what they are, but gets the independent variable. So this gets hard when we're looking at things like global warming. It gets hard when we look at things like changes in the ozone layer, because those are hard to replicate in a lab. Drug study is fairly easy for me to explain to you, but we do try to replicate those sorts of phenomena in a small scale in a lab. So maybe we would apply the chlorofluorocarbons, I talk about it in your book, from aerosols to see if that impacts the chemical ozone, because we have that in a in a canned setting. We can actually use that, and then we can look to see if that erodes the ozone, and then kind of extrapolate to an increase into what we see in the atmosphere to changes in the ozone hole that's there, and the sizes, which are measurable. It's just more difficult when you're looking at the whole Earth versus a drug study, and just kind of making that comparison. And sometimes we have some experiments are simply observational. And sometimes we see that in education because it's sometimes maybe considered, counterproductive, not morally the right thing to do is to say, oh, we're going to give these students access to technology and these students not. You don't want to make some of the students not getting some of the things the other ones get. But because those differences do exist within the education system among school districts and schools, you can actually just observe and measure that. So you just kind of measure what is in place. There's also things like lifestyle. You can look at people who have different lifestyles over the years. So sometimes people who study that will go to sometimes a small island or a religious group that has very, very specific lifestyle practices, and they'll look at the overall health, stress levels, cardiac, age span, things like that. So sometimes we're not always running a quote-unquote experiment. And then the last thing I wanted to mention here is the dependent variable. And this is the measurable change. And this is one that students get confused with the independent variable. So it depends, I'll make this all caps here to really emphasize my points, depends on the impact of that independent variable. Did the drug lower blood pressure? Did it change cardiac health, that sort of thing? So caffeine is an example you can use. Does that affect heart rate? So the change in that case would be, and this is again looking at a drug study, might be symptoms of a disease, so pain, things like that, which again can get difficult to measure because that's can be a little subjective. So things like symptoms, you can look at blood pressure if you were looking at blood pressure drug, heart rate. Lots and lots and lots of any medical measure you could think of could work for certain drug types that are there. And then lastly, I want to talk about theory, because sometimes that word gets misused a lot. And so I think they do a nice job in the book kind of explaining the difference between those. So I would commonly say, well, in theory, I'm going to be able to get across town in 30 minutes. And I use that because I've been going across town a little bit more this semester, so it's on my mind. But for scientists, this is something that has a high degree of repetition many times when an idea or conclusion can't be supported by subsequent experiments, and they talk about trying to duplicate or replicate that work, then it wouldn't become a theory. So it's a very high threshold for these scientific conclusions to become a theory. It's not a quick jump. It takes many years, many different groups being able to replicate that work that's there. But we use the word in theory in just day-to-day vernacular. It means a little bit something different. It's really more of an idea. Well, in theory, I think, but in a scientific theory is much more concrete and has to be very heavily supported. So, when you have a theory that's there, there's always that remote chance that you're going to toss it out for some reason. And so your book gives some neat examples. They have theories that have held, like the gene theory, theory of evolution, cell theory. So we've never found a living organism, for example, that's not made of a cell. But what if we did suddenly find living organisms that weren't made of cell? That would throw that into question. However, hundreds of years that's never happened, so we're not expecting that will. And the same is true of other theories that you hear out there. Now in physics, sometimes they go all the way to law because those are so replicable in different experiments that are there. But there are theories that have been thrown out over the years. And your book gives kind of a fun thing at the very end where they talk about there used to be a theory that the earth was flat and that the sun circled the earth and not the other way around. So as we got more technology, more measurements, that wasn't supported, so those were cast aside. So hopefully this gives you a little bit of help with the scientific method and was good use of your time. So I appreciate you listening to the lecture.